Hey, Bio 30. This is our first lesson on 16.1. We are starting our genetics unit, and we are going to start with chapter 16, looking at cell division, but we're going to kind of make our way around what DNA is all about to start with. Remembering in this picture of an animal cell that DNA is found in the nucleus here. If the cell was dividing, the DNA might look like X-shaped chromosomes. But if the DNA is not, or the cell is not dividing at that time, you would just see the DNA as these strands of chromatin, and they would organize themselves into X-shaped DNA just before the cell carries out cell division. And that's what we'll be talking about in this chapter. This little launch lab question asks, how in the world can you get a 16 chromosome cell to make 2 copies of itself and have 16 chromosomes in each of the 2 daughter cells? You can check that little launch lab question, but to answer it, basically what happens to every one of these 16 chromosomes is that it will make a copy of itself. That's why it looks like an X, because it will make a copy of every chromosome, and those chromosomes that then look like an X will separate so that each of the daughter cell will get half of that identical 2 parts of the chromosome. So all of the original genetic material found in our original cell will still be in each daughter cell. So it'll still have the same number of chromosomes. This one had 16 chromosomes. Each of these will also have 16 chromosomes because the X is split in half. That's kind of a preview of cell division, where body cells make copies of themselves. It's called mitosis. We'll be talking more about that later. There's 4 topics: introduction of the cell cycle, how body cells divide, which is mitosis, how we make sperm and egg cells, which will be meiosis, and then a little bit about some reproductive life cycles in 16.4. So to start with, you can check out the dinosaur chicken video. Cell size is something we've mentioned in Science 10 before, that as a cell grows, the cytoplasm will also grow. However, there is a finite amount that cells grow. So cells are all very tiny because as the cell grows, their surface area to volume ratio goes down. And when it's a tiny cell, there's a lot of surface for diffusion of oxygen and nutrients to enter a cell. If it gets bigger, there's less surface compared to the volume. If it gets too big, there's even less surface. As you can see, it's 1.5 to 1 compared to 6 to 1. Way less surface compared to all the volume. And basically, it takes so long for the oxygen and nutrients to get into the cell and even get all the CO2 out of the cell that it's not an efficient thing for the cell to do is grow that big. So you can see that cells are typically all kind of in the similar, like small, very small cubic micrometers, and the biggest one that we have that we know of are human egg cells, which is 4 million micrometers, cubic micrometers, and that means like if you had the tiniest like speck of dot that you make on a paper, your egg cell might be visible to the naked eye. Otherwise, all cells are much smaller. The egg cell contains a lot of cytoplasm and is the biggest cell that we know of. This demo you may have seen before with cubes of Jello, because we do it, I've done it in grade Science 10 as well, but we take these 3 cubes, make them bigger and bigger and bigger, and basically they're a red, made of a red dye that will turn yellow when an acid touches it. So we dip these red cubes of Jello into a solution of hydrochloric acid and look at how quickly the diffusion happens because it turns yellow. So if you remember, this one will turn yellow the fastest. This one, you know, the edges, it'll get there. This one much slower. And of course, it'll take this one much, much longer to diffuse and get anything right into the center. So it's just confirming that it's not very efficient to have cells be too big. Okay, looking at parts of the chromosome, I'll just point out this chromosome at the bottom here that you can see is our X-shaped chromosome. And what we want to look at is how the DNA is arranged in there. And basically, the whole idea is to realize it's folded many, many, many times over. So DNA, deoxyribonucleic acid, is kind of this double-stranded, twisted molecule that's got the code for basically the recipe that determines how to make all your cell materials, body parts in your body, how to make all your hormones, all the things that your body makes, enzymes, everything. All the instructions are in the DNA, which you'll hear all about very soon. And it's a very fragile thing, so it's wrapped around these little histones, these little protein molecules, to give it some structure. And I can, I guess, relate that to maybe wrapping your saran wrap around a cardboard tube. If it wasn't wrapped around a cardboard tube, it would be a mess because it's so fragilely thin. What happens then with the histones is it gives it some structure to wrap the DNA around. And the chromatin is the big strands of DNA, and those chromatin fibers are all wrapped around those histones and then folded. So we fold this guy over here, we fold it even more and put it onto the piece of protein scaffold. It's like standing on a scaffold again to give it some more structure. It's folded and it's on a piece of protein, and then it's folded again. So it's just showing you how DNA is wrapped, folded again, and found in this chromosome and organized in your cell nuclei. There are 46 of these chromosomes in every body cell that you've got, which is 23 from your mom and 23 chromosomes from your dad. Those were the original ones found in the sperm and egg cell, and made a set of 46 chromosomes, and every time cells divided, every cell will have a copy of those same 46. So any cell in your body will have all the information to make all of the body parts that you have. You guys have this picture in your notes of a chromosome showing you the loops of chromatin in there, how they're wrapped around the histones. And if you look at just the double-stranded, double helix DNA, that's what it looks like there, and you can fill that out. The structure of a chromosome is that when you see a replicated or doubled chromosome that's the shape of an X, these could be called sister chromatids that are identical. And in the center, it's held together by a centromere, which is just it's a type of protein that holds it together so that chromosome doesn't separate until the cell division process is underway. So it just keeps it very organized to hold the two sides together. This is a picture of an actual microscope picture of a chromosome. A gene is a section on that chromosome, and it's a piece of, if we just look at this section right here, it's a section of the chromosome that's got the recipe on how to make a certain protein. And it's like having a recipe book, and each section of it has a recipe for a different thing. So this might be the recipe on how to assemble an insulin molecule, for example. And so within our 23 chromosomes, because the other 23 are from the other parent, but within those 23 chromosomes are all of the recipes to make all of the organs, cells, and cell products, hormones, enzymes, all those things that are found in your body. Okay, now chromosome number is not the same for all organisms. For example, we know that humans have 46 chromosomes. Lettuce appears to have 18, chicken 78. I put this in PDF in your in Google Classroom, so you'll be able to see the whole thing. But it does vary. What we do know is that we have, I'll just show you a karyotype here, that in humans, our 23 pairs mean that we've got them paired up, so we have they kind of line them up longest to shortest, and then the sex chromosomes are last. But we have 23 pairs, number one from mom, number one from dad, number one from mom, number two from mom, number two from dad. And so when you pair them all up like this and make this karyotype, a sort of arrangement of your one cell's worth of chromosomes, you can see that the last pair of chromosomes is the sex chromosomes, meaning that if they're the same size, that would be considered two X's, and that would be considered female. And if they are not the same size, one is an X, one is a Y, that would show you that that is a karyotype of a male. So that's just showing you what 46 chromosomes look like in a human. And we've got some examples here of some other organisms that have different chromosome numbers. I guess just to say here, the word allele, that is the version of that gene, showing you that not both the chromosomes, one from mom and dad, necessarily carry the same version. So one may give you a gene for black hair, one may give you a gene for blonde hair. So the different versions of a gene are called alleles. And if you have two, let's say number 6 chromosomes, you will see that at the same location you have the recipe for that exact gene, but it may be a different version. So the alleles are just different versions of that gene, maybe different colors, maybe different heights that the plant can grow or whatever. Okay, so the sperm and egg carry a haploid number or half number of chromosomes, and a body cell would be carrying, or a somatic cell or body cell would be carrying the double number of chromosomes, as you saw in those karyotype pictures a second ago. And so we write down the gamete chromosome number as represented by N, just means the haploid number, and for humans it's 23. And so it means every body cell would have the diploid number, which for humans is 46. Some organisms are kind of crazy and have multiple copies of their chromosomes. And so in our next couple slides here, we're looking at some organisms that have 3 copies of all the chromosomes. So if you had a karyotype of them, it would be like 3 number 1s, a little bit shorter, 3 number 2s, 3 number 3s, 3 number 4s. So that would be what triploid organism would be like. For example, seedless watermelons. Tetraploid salmon, a certain type of salmon species, has 4 copies of every chromosome. We've got some birch tree with 5 copies of every chromosome, wheat and kiwi with 6 copies of every chromosome, and sturgeon and dahlias with 8 copies of every chromosome, octoploid, and decaploid, 10 copies of every chromosome in strawberries. So that sounds pretty nuts, but it doesn't stop there. There are 12 sets of every chromosome in this Uganda clawed frog. So although he looks kind of to be a, like regular little cool little guy, he's actually got a pretty crazy looking karyotype. If you had to work with his genetics, 12 copies of every chromosome. Cool stuff. And we find that this fern also looks kind of like a regular little thing, but it has 1,260 chromosomes in every body cell. That would be nuts to work with. And in the animal world, we've got this rat that the number seems to have disappeared on, but in your PDF, I'm sure it's there. Anyways, that wins the prize for the most chromosomes in every body cell here. Is this crazy little rat. And remember, your body cell has only 46 chromosomes, and this guy, I will have to check that out as to what it was, but it is definitely way more complicated than you. And I'm just checking this out here. It appears to be 102. Number just went away. So 102 chromosomes in every body cell. Okay, those are just some cool examples. No need to memorize those or anything. Karyotype is a picture of your 46 chromosomes, all organized in its with its homologous pair, meaning the other one that's carrying the same chromosomes. Those are all arranged with their homologous pairs. And if it has two sex chromosomes that are X's, it's a girl. Sex chromosome that's one an X and one a Y, that is a boy. These are actual electron microscope pictures of a female and a male sex chromosomes. By the way, this still looks like an X. It's just a shorter one. And this is just showing you what those look like. Okay, to organize a karyotype like we see there, you would have to stop the cell from reproducing at the point at which those X's are visible, and then photograph them, and then arrange them longest to shortest with its homologous pair, and put the sex chromosomes last. So that's how a karyotype would be organized and arranged, and they would do this to see that there are only two of every pair, because if they have an extra one, for example, number 21, if there was an extra 21 there, 3 21s would indicate that this individual will have a Down syndrome. And that's how they check for genetic disorders, is when they check that the chromosome numbers are correct. And for the Down syndrome, having an extra number 21 would actually give that person 47 chromosomes instead of, in every body cell, 47 chromosomes instead of the 46 that are usually in every body cell. So we'll talk more about genetic disorders a bit later. The last thing for this topic is talking about the cell cycle and how 90% of the time the cell in its lifetime is going to be in what we call interphase, which is preparing for cell division, and 10% of the time actually dividing and carrying out the stages of cell division called mitosis. And so in those 90% of the times of interphase, the in-between phase, it will do the G1, kind of referring to growth, or gap 1 phase. And this is where the cell is growing body part or cell parts. And then it will do S phase. Synthesis means forming something, and it's actually going to be forming the other side of the chromosome and doing replication and doubling itself. So in the S phase is where all of the DNA is going to double, and you still have your 46 chromosomes, but they just double and form the other side in preparation for cell division. And for the gap 2 or growth 2 phase, it's just going to continue growing some more parts of its cell. And after the G1, the S, and the G2 phase, it is ready to do cell division. So in this 10% of the time, the cell division process that's happening there is the phases that we're going to talk about in 16.2, which is the, can't see that, the PMAT stages. And those stages of mitosis are called prophase, metaphase, anaphase, and telophase. So we'll talk about that a little bit more coming up, but you guys just have to know that there's these 3 stages, G1, S, and G2 in the interphase. You know then that 10% of the time it's actually going through mitosis or cell division. And the word cytokinesis is that splitting of all the organelles. If your cell is splitting in half, all the organelles are split in half, and the cytoplasm is split in half, and cytokinesis is the actual splitting apart. By this time, half the chromosomes, they would not look like that. They would look like this. Half the chromosomes would go onto one side, each chromosome would go on the other side, and cytokinesis is when it actually splits all the cytoplasm in half. So they have researched this many times to realize a cell has only so many times it can split in half. In human cells, tends to be about 50 times that it can do cell division before it dies. They've tried letting it divide 20 times, freezing it and defrosting it, and it still divides 30 more times. So they can't seem to change that. It means that cells have a lifespan, and so that's why humans, as a rule, generally have a lifespan in and around 100 years-ish. And that's because cells can only divide so many times. The word apoptosis is the programmed cell death, and what that means is that sometimes there are cells that are actually programmed to destroy themselves. So examples of apoptosis, if you check out the link, are things like in fetal development, the little hands and feet look like little paddles, but there's going to be cell death to form the thumb and all the fingers, and all the cells in between will die in order to have all the digits of your hands show up. And then other examples are little tadpoles that have tails. There's cell death that will get rid of the tail. It doesn't just fall off, it's just cells that die. And another example is our menstrual cycle of the uterus. So the endometrium there that is shed is cells that will are going to have apoptosis going on, and they're pre-programmed cell death. So there's some examples of cells that that have only a certain lifespan. Alrighty, that is our 16.1. Thanks for listening, and I will